Hi, Ellen Donnelly, CEO of Abliva. We're here for day 4 of Mitochondrial Disease Awareness Week. In our portfolio, we have 2 important assets, NV354, which you heard about earlier this week from Eskil, and KL1333, our lead program. KL1333 will start a phase 2-3 study later this year, which, if successful and everything happens on time, could be on the market by 2026. Today you'll hear from Magnus Hansen, our Chief Medical Officer, about the KL1333 development program. Thank you.
During this series of videos, we've stressed the high unmet medical need in primary mitochondrial diseases. There are also other companies working in this space. Could you briefly tell us about that?
Yes, there are a handful of strategies being tested by different companies, most of them with different mechanisms of action. A couple of companies aim to improve the energy metabolism in the cells, similar to what we do at Abliva, but they target other and potentially complementary pathways. Others focus on improving the cognitive aspects of the disease. Abliva is the only company, as we are aware of, that has a sole focus on the development of therapies for primary mitochondrial disease and has a full portfolio of programs.
Abliva has a real opportunity here to contribute meaningful therapies for primary mitochondrial disease patients. KL1333 is Abliva's most advanced project in the portfolio and will start a registrational study later this year. How does KL1333 work?
KL1333 is a new type of compound specifically designed to improve energy production in mitochondrial disease. The aim for the molecule is to improve energy levels throughout the body's cells so they can function properly. KL1333 does this by normalizing the balance of NAD, which is a key molecule in the cell. When mitochondria don't function properly, there is a buildup of one type of this molecule, called NADH, and a lack of the other type, NAD+. And this imbalance obstructs the normal energy production. By restoring the balance of NAD, KL1333 boosts energy production, both short-term and long-term, by stimulating buildup of new mitochondria.
A number of studies with KL1333 were completed earlier, including a phase 1b trial in primary mitochondrial disease patients. What can you tell us about those results?
In general, the numerous studies we have completed have given us a good insight into the general properties of KL1333 and helped us identify the right dose range for the phase 2-3 study. The most exciting results came from the phase 1b trial in primary mitochondrial disease patients, where we saw improvements in those treated with KL1333 compared to the patients who received tablets without KL1333. One important aspect of this study was that neither the patients, nor doctors, nor we knew who received which until after the study was completed. Improvements were seen in both muscle strength and fatigue symptoms. And in fact, the patients who obtained the highest concentrations of KL1333 showed the greatest improvements.